Hello everyone, welcome back to the Master Food Tech Academic Concepts Made Easy. In today's video, we'll solve the 10 very important MCQs which can be asked in any competitive exams. So let's start with the first question. The pH of fresh milk is approximately. So these are the pHs they have given. Before going into the options, let's see the concept. Fresh milk, it's like a slightly acidic to neutral in nature, right? Acidic to neutral in nature. And the normal pH range of fresh cow's milk would be 6.6 to 6.8. And this acidity is mainly due to the dissolved carbon dioxide and the phosphate and citrate buffers and the milk proteins. If the pH goes below 6.6, if the pH of the milk goes beyond below 6.6, it indicates that the microbial activity or spoilage is due to the lactic acid formation. Because the fresh milk pH should be nearly acidic, like 6.6 to 6.8. If it goes below 6.6, it means the formation of acid is more, which is lactic acid formation is more, and thus the spoilage of milk happens. So the correct answer will be 6.6. Next question. The critical control points concept belongs to. So I guess this might be little easier question. CCP. In the sense critical control point, which is a core concept of HACCP, hazard analysis and critical control point, right? Which is a preventive food safety system, which is not a testing system. It's a preventive food safety system where a hazard can be prevented or eliminated or reduced to an acceptable level. So here examples of HACCP we can say in case of milk, dairy industry, their pasteurization temperature is one of the critical control point, and even the metal detection, even chilling temperature. So these are few critical control points which you have to check after in a dairy industry. So critical control points are related to HACCP, hazard analysis and critical control point. So what are the prerequisites for this HACCP? Prerequisites are GMP and GHP, good manufacturing practices and good hygienic practices. So here CCP belongs to HACCP. Next question. Which microorganism is used in yogurt production? So these are few microorganisms they have given. Which microorganism is used in yogurt production? So yogurt is produced by the lactic acid fermentation of milk, right? So the starter cultures used here are Lactobacillus bulgaricus and Streptococcus thermophilus. These are the two main important microbial species which are used in the production of yogurt. So what does this microorganisms do? They convert lactose to lactic acid. So for any So even in milk, for the formation of curd also, lactose will be converted to lactic acid. Then only we'll get curd, right? From milk. So here the major role, uh, which does the major role is microorganism. Which kind of microbial species are used for the conversion of lactose to lactic acid. So here the microorganism used is Lactobacillus bulgaricus and Streptococcus thermophilus. So these are the both important microbial species which are used for the production of yogurt. So here the answer would be Lactobacillus bulgaricus. Moving into the next question, blanching is mainly done to. So before going into the options, let's see what is actually blanching is. Blanching in the sense it is a mild heat treatment usually done before freezing or any drying process. So it is a mild heat treatment. Mild heat is given to the food prior to freezing and drying. Freezing or any drying. So what is the main purpose of this blanching? It is especially used to inactivate the enzyme. So enzyme inactivation and to soften the texture of any food. Okay, soften the texture like surface area. Next is to partially cook any food item, right? So this is a pre-cooking method, pre-cooking step, which we have to do in order to achieve these things. Enzyme inactivation, soften the texture and partial cooking. So what kind of enzymes it can inactivate? Polyphenol oxidase, peroxidase, catalyze this kind. If enzymes are, why these enzymes need to be inactivated in the food? Because if these enzymes are not inactivated, they can cause browning, right? Browning of the food or cause any off flavors, or even we can lose the texture loss. So these are the advantages of enzyme inactivation. So even the basic thing which we'll see here, like blanching any brinjal or cauliflower, these kind of things, in order to prevent browning. Before cooking, we used to do in kitchen, right? So this is the main basic principle behind the blanching. But most important thing you have to look after is blanching does not kill the spores. So this would be the wrong. Even removing the moisture, improving the color, no. The enzyme inactivation is correct. So next question. So moving on to the next question. Next is water activity. Water activity range for the bacterial growth. What is the water activity range for the bacterial growth? So what is water activity? Water activity indicates the available water, which is the free water available for the growth of the microorganisms, not the total moisture content. Right? Moisture content is different and water activity is different. Water activity in the sense it is the freely available water present for the microorganisms to grow, right? It is not the total moisture content. So bacteria require high water activity to grow usually. When compared with the moles or yeast, bacteria require high water activity. So 0.9 would be the most bacteria grow at this water activity. Next, yeast usually grow around 0.88 and moles 0.7. So that's why dry foods are safe. Even the high moisture foods spoil faster, right? So for bacteria, always remember water activity should be greater than 0.9 to grow. Next question. Which packaging material has the best oxygen barrier? So for any packaging material, there would be layers of the packaging around one of the packaging, right? So for which material? So this can be LDP, HDPE, or anything, right? So which is the best for the oxygen barrier? So here aluminium foil is an absolute barrier to oxygen, light, moisture, and even gases. So aluminum foil is the best. Plastics like LDP, HDPE allow the oxygen permeability, right? That's why aluminum foil is used in the retort kind of pouches, chocolate wrappers. Because for the chocolate, there shouldn't be any exposure to oxygen. So, in order to prevent the exposure of oxygen or permeability of the oxygen into the packaging material, the chocolates also use the aluminum foil. Next, pharmaceutical packs also. Pharma. So the best barrier, oxygen barrier would be the aluminum foil. Next question. Smoking of fish primarily helps in. So before entering into the options, what is smoking actually? Why it is done? Smoking is usually both preservation and flavoring method. Preservation, to preserve the fish, smoking is done, and to enhance the flavor, smoking is done. So it helps by depositing the antimicrobial compounds. Antimicrobial compounds are deposited on the surface and even helps in partial drying, antioxidant action, giving the characteristic smoky flavor. So, most important, smoky flavor is given. Smoking does not increase the fat or absorb the moisture. So, this is the advantage of smoking. There would be disadvantages also because they can provide carcinogenic substances also. Next is, so the correct answer would be the flavor and preservation. Moving on to the next question. The process of separating cream from the milk is based on. So usually cream separation, it works on the principle of density difference. There should be some density difference, right? So milk fat is lighter than the skim milk because milk fat is lighter than the skim milk. So using a centrifuge, heavier skim milk moves outward. So centrifugal force centrifugal force is applied when centrifugal force is applied in the centrifugation, in the sense the force moves against, right? The force moves outward and the lighter cream moves inward. So here skim milk, watery particles moves outward and the fat particles moves inward, and thus this process is called centrifugation, not filtration or sedimentation or evaporation. Centrifugation. Through the process of centrifugation, milk is separated. So moving on to the next question, which vitamin is destroyed by heat easily? So you have to remember one thing is vitamin C, which is an ascorbic acid, is heat sensitive. Heat sensitive, oxygen sensitive, water soluble, right? And during cooking, blanching, or pasteurization, vitamin C losses are very common because it is very heat sensitive. Fat soluble vitamins like A, D, K are more stable to heat. High stable to heat. So not A, D, K, vitamin C is heat sensitive and thus it can be easily destroyed by the heat. Next question. Codex Alimentarius is related to. So what is Codex Alimentarius? Codex Alimentarius, which is it is a collection of international food standards. International food standards. As for India, the food standards will be FSSAI, right? Food Safety Standards Authority of India. For the international, there would be Codex. Codex Alimentarius, right? And it is jointly developed by FAO and WHO, Food and Agriculture Organization and World Health Organization. So it helps in consumer safety, fair trade practices, and the harmonizing the food laws. So remember, Codex means it is a international food standards. So these MCQs are very important for GATE, Excel or XC or any other competitive exams. If you found this lecture useful, kindly like, share and subscribe to Master Food Tech channel. So more MCQ based and concept based videos are coming soon. Thank you and happy learning.